Hello, everyone. Thank you for staying late. And please allow me to switch the gear a little bit and from the human research, but I hope that it will still be interesting to you. I also want to thank the committee for choosing our paper, and I also take this opportunity to thank the three reviewers for your very constructive suggestion for improving the manuscript. So this work I'm going to share with you represents mostly computer science master thesis by Andrew Wu under the close supervision of myself and my colleague Ethan Lee, who is a computer scientist, but also expert in AI and machine learning. And this is a part of a big new project that I'm responsible. But I have to say that I know very little about machine learning and AI. It happens that either of them could come to this meeting, so I'm the only one available to present this work. So I'd like to give you a warning or a disclaimer. I can try my best to explain the background, but not too much on the machine learning part. All right, okay, so just a quick outline. I'm gonna just give you a brief history or importance of the grapevine and the need of genetic identification, genetic testing, but also a brief summary of this, the technology or the methodology and the state of the art, and then move on to the project we are working on, focusing on the CGR combined with deep machine learning. All right, so as probably many of us know, grapevine is a very important crop for many parts of the world, especially for like Southern Europe, here in the States for California, right? And from where I am, Niagara Peninsula, now we actually have gained the title of wine country in Canada by having over 200 winery and over 13,000 acres of vineyards. And we are very famous for ice wine and being one of the largest ice wine producer in the world. In case you have never tried ice wine, I would highly recommend you find the opportunity, excuse for yourself to taste ice wine to enjoy the amazing aroma and very sweet taste. Anyway, okay, so grapevine actually is one of the crop that has the longest history of domestication. For those ones that, if you are familiar with Bibles, the first recorded viticulturist is Noah. That's recorded in Genesis chapter 9, verse 20, I believe. And so with such a long history, a lot of variety has been accumulated. So minimally, we have over 10,000. And this morning, I looked at another database. It says 23,000. And that's a great thing, right? But it also, over the history, such a long history, there are a lot of names. There's no standard of naming grape cultivar. You can name anything you want. So what end up happening is there are a lot of synonym, which means same name, well, different name for the same thing, or homonym, it's the same name for different things. One extreme example of that is this very popular cultivar called Greenwich has over 100 synonyms. So that actually leads to a lot of confusion and errors. And so when it comes to the variety of grapes, there's three levels. One is at the species level. Most of the species we have, or exclusively, are from the Vitis genus. and we have hybrids across different species. But when it comes to cultivar, it's the variety within the same species. It's mostly from Vitis vinifera, and we have lots of them. The vinifera variety constitute over 90% of all the variety we have. And then we also have clones for some of the very popular vinifera cultivars. We also have clones that have very subtle differences. So those represent different level of challenges when it comes to identifications. And identification, being to accurately identify a specific species or cultivar or clones is very important for a lot of state owners, such as nurseries, wineries, and even consumer like us. We want to know what we're drinking is what is claimed to be on the bottle. All right, so that talks about the importance of the genetic identification, what we call type, depending on who you're talking to. So traditionally, until we had this genetic molecular biology markers available, the only methodology available is something called apolographies, which came from a Greek word, basically means the writing or joining of wines. And it's mostly based on the morphology of the leaves, but also some other things like the shape of the cluster and the color of the grapes. But of course, the morphological difference is limited to some degree. So, and it's also subject to human errors and also environmental factors. So, of course, there are limitations to how much you can do. And not too long ago, there's an attempt to use the machine learning to improve the accuracy, but still, I mean, it's limited by what it is. So, and then I guess we had the genetic testing, starting with the use of sequence tag sites, but the test wasn't available as a commercial service until like year 2000 with the use of the short sample or tandem repeats. Okay, so this is very similar to what we know for human DNA fingerprinting. It's based on the size set of markers, and we look at the size difference is a database. And then the main issue of that, the limitation of this approach currently is there's a very limited service available. Around the world right now, there's only 4 or 5 different places. For people like us in Canada, the closest is the one in California. They use Davis. And it still costs more than $400 per sample, let alone you have to pay international shipping, dealing with all the paperwork for shipping the materials across the border. So it's there, but it's very limited, and it actually could be quite expensive and painful to use. All right, so now we are into the era of next generation sequencing technology, and that presents a new opportunity for doing the Grapevine genetic testing. And so there's one group with the traditional method. They try to use the NGS to sequence the PCR amplicon for those markers, trying to see whether they can improve the cost efficiency or accuracy. It does improve the accuracy by not only having the length of the products, but also have the sequence of the products. But again, it's still limited to what markers we have. It's only a very small set of the genome, right? A portion of the genome. And the cost is actually end up being higher than the other one, the traditional one. So we are looking into developing a whole genome-based sequencing approach. And the rationale behind is now the cost of sequencing the whole genome is becomes so cheap. And based on my calculation, we can actually get to about $200 for sequencing, and plus the other overhead, we can probably make it cheaper, if not, or at the most is going to be about the same as the traditional method. There are several other advantages, right? In theory, by having the whole genome sequencing, we should be able to really distinguish any individual plants, right? And also, if you do genome testing by doing the whole genome sequencing, the customer would actually also have a copy of the genome sequencing available for any possible future genetic studies. So there are a lot of advantages. So the goal here is really trying to make the test more accurate, more available, and then more affordable. I mean, yeah. But one thing is, in this case, nowadays, because the sequencing service is available everywhere, so in this case, the customer would have an option to either send us the sample and we do the sequencing, or they could actually do the sequencing and just share the sequencing with us. So, all right, but of course, dealing with the whole genome sequencing is always computationally challenging. Another part of the rationale for doing what I'm gonna present to you is that we already, in the public domain, we have already accumulated all of the whole genome sequencing data for Grapevine. And so at this point, it has a wide coverage, over 40 different species, some hybrids, and over 3,000 vinifera cultivar. It's mostly available at the SI database as NCBI, and recently several Chinese groups also did a couple of large-scale Grapevine sequencing. And we also are in the process of generating our own locally sequenced data just to supplement what's insufficient in the public domains. All right, so there are limitations to those data. They are mostly generated using the Illumina short-read platform. And so it's hard to get the high-quality genome assembly. And yeah, so even with the assembly, whole genome assembly, it will be difficult to do direct genome level sequence comparison. So our approach is to go with the whole genome raw data, Illumina, for example, and we go for variant identification, and then we just go with the variant. Yeah, looking at the variant, so that reduces the size by 10 to 90%. And also in this case, the good thing about that data set is most of those samples have their cultivar or species assignment already. So this information serves as a gut choose for whatever test we're going to be developing. All right, so what we have tried so far is several methods of analyzing those whole genome WGS data. One of them is just the k-nearest labels. In this case, we're just comparing the variant sequence similarity and then trying to cluster them. But the results are not really great. And I also myself tried the k-mer approach. This meant to be a relatively quick and less computational demanding. I mean, not for memory. It actually used quite a lot of memories. And it actually turned out to be not that great either, although it actually performed well for one of my own data set, which has very homogeneous coverage. So this really motivates us to look for a novel approach to analyze those data. and the approach one of the approach we took is the CGR with the machine learning, which is what I'm going to tell you. Just briefly, just in case you don't know about CGR, it's graphic visualization. It's a chaos game representation, and it was pioneered in the 90s by Jeffrey as a square representation to represent the 4 nucleotides of the DNA instead of the triangle, which is the original CGR format. And by mapping the next space between the previous space, the middle point of the previous space to the next space, you can actually generate a picture, image like what is showing at the top. So this is actually the whole chromosome 1 sequence of one of the species, grape species. And if you just use the variant, then it's much less denser. And with the upper one, you could easily, depending on the resolution you're using and how long the sequence is, you could easily oversaturate it and then lose any discriminations. So in this case, it converts a 1D DNA sequence into a 2D image. And in this case, the sequence could be variable in length, but the images the size is determined by the resolution, so it can be equal size. And it is important to note that this conversion is not lossless. Okay, so there is information loss. But the great thing is, once you convert it to an image, it allows us to use all kinds of machine learning algorithms. All right, so to prepare our data for this kind of process, we picked some of the data among all the over 4,000 data sets, and only choose the one that meets certain minimum requirements. And so we also run the variant identification. We also have sort of a threshold, what to encode. But in the end, all the sample will have the same number of variants, loci in the sequence. And then we convert that into a pseudo sequence. So those sequences are one-to-one corresponding, orthologous corresponding. So I think that's very important. And by just focusing on the variants and also converting to the CGR image, we can actually reduce the data size by 10,000 to 50,000. All right, so just for the training of the model, this is how we actually divide our sample for each class. It's 70% for training, 10% for validation, and then 20% for testing. That actually eliminates many of the classes because the sample size are very variable. And for some of these samples, like vinifera, at the species level, we have several thousand. So we have to actually limit it to about 60. And then so in the end, we have, for species level, we have 19 different classes ranging from 3 to 60 sample. And for culture level, we have 163 classes ranging from 3 to 7 samples per class. And then in this case, because it's a genome, it's assigned to individual chromosome. We have a total of 19 chromosomes. We can treat them as individual chromosome, each chromosome as a separate entity, but we can also combine the sequence as one per genome. And that actually is something we look at it more later. All right, so there are a lot of things to be optimized or to be tested and optimized. In terms of resolution, there's those after many testing, this is what Andrew came up, 3 different resolution, and then different way of normalization. They want that in the blue color, highlighted in blue color, the one that we end up using. And then I will leave out the other details for you to look at it. But for integration method, there are 4 different methods. So one is just to treat each individual chromosome individually and do all the training and testing. But the other one is to train in each individual and then combine the model, and then using the combined model for later stage analysis. Or we can try working on individual chromosome and then in the end, just using a voting system to among their 19 chromosomes. And then the last one is just to combine the whole genome, all the 19 chromosomes as one sequence. All right, so we also implement the fine-tuning. Actually, we tested all those different models available for us, and then we ended up focusing on the RASlet and the VIT, two of them, for more extensive testing. And yeah, so we came up a strategy of how to assess in the end the performance of each model. So just to give you an example, this is the result for species level classification. And I thought I had to explain what this plot is, but I saw Shelfie use this spider-led model. So here, basically, we have 3 plots showing you different resolution and with different way of the integration that I just explained it to you, and then combined with either with normalization or without normalization, but also serving either 100% or 50% or 25% of the classes, okay? And as you can see, the overall pattern between the 3 resolutions are, overall pattern is more or less the same, but details are different. So as you can see that if we focus on the one on the right, the best performance is the one for 25%, top 25% by class size. I will explain that to you later, what this means. And also among these 4 different methods of integration, the late integration outperforms. Okay, all right. So and then this is the result of the cultivar. I have to point out the scale is not the same, but yeah. Just for the sake of time, I will just show you this 2 table. This just shows you the two methodology, 2 models, and as you can see, the RASlet consistently performs better than VIT, and in this case, this is the species level for 25% classes. And on the other side, down the right side it shows you a summary of the performance. So in either case, all the machine learning method capture significant signal over the random match. And then here the RASnet performs better for best close to 100% at the species level. At the culture level, it's not as good as we wanted, but we know what to improve. All right, just to, maybe I'll add this as a quick summary. So machine learning gave us some encouraging results, and we actually figured out a lot what will work, what doesn't work. And then one critical thing tells us is the class size. Why top 25% performs the best is because it has larger sample size per class. And so this gave us a good direction what to improve in the future. All right, so, and then we also have many directions for future work. Just for the sake of time, I'll stop here, and while I take any questions, I'll leave this slides with you. Well, I think the people did the work, and then also the funding agency. Yeah, this is the slides I want to leave you while I address you any questions you may have. Thank you. One or two questions. Oh, thank you.
Yeah, thanks for the impressive work. So I'm curious for the chaos game representation, since it's I think it's like jumping the dots in the graph, like from if the genome is A, G, C, then you jump to several points in the graph. So it feels like after several points, you will lose the resolution. So in the end, the graph is from the k-mers or from the genome?
Yeah, so this is part of the issue with CGR is to some degree, you lose the physical connections, relationship.
So
yeah.
we will lose that.
Yeah, you will lose to maybe, I mean, the exact degree of loss, I can't tell you, but the loss is there.
I see.
Yeah.
Yeah. So another question is for the for when you call the variant. So I'm not familiar with Grapevine reference genome. So is there a reference genome for that, or there's a reference genome for different Grapevine species, and then you do the alignment to them and then consolidate the variant calls?
Yeah. So for this data set where I showed you is using the vinifera references, but now we have pan genome available. Yeah. Yeah, it should be better. Thank you.